Welcome back to another episode of DNA Unsolved. This week we'll be addressing the controversial question, are we related to monkeys?
Let's get to it.
On July 21st, 1925, substitute high school teacher John T. Scopes was put on trial for teaching human evolution in a state-funded school, which was against the Tennessee Butler Act.
Although it was a simple case of whether or not the law had been broken, it grew into a major clash between religion and evolution. The trial was only one of the many instances of a dispute between religion and science, but this time it occurred specifically in response to evolution.
Today we are going to try and crack the controversial question and figure out once and for all whether or not we're related to monkeys.
Our theory brings about a more scientific approach and states that all organisms are subject to evolution, including humans. More specifically, the theory states that all humans are related to the great apes and share a common ancestor with them. This idea can be traced all the way back to the publication of Charles Darwin's On the Origin of Species. In his book, Darwin explained the theory of natural selection and how it led to the rise of new species. This idea of evolution through the mechanism of natural selection has become one of the most widely accepted theories among scientists today.
In fact, the theory has been backed up by extensive genetic and chromosomal evidence and is therefore almost unanimously supported by the scientific community.
The first piece of evidence is provided by the analysis of chromosomes 2A and 2B of chimpanzees and chromosome 2 of humans. While great ape species have one more chromosomes than humans, all researchers arrived to the same overall conclusion, that every human chromosome, except one, has a matching chimpanzee chromosome with striking similarities in sequence and organization. Human chromosome 2 is the exception and matches with two chimpanzee chromosomes, 2A and 2B.
It was in 1991 that scientists of Yale University found a DNA sequence in the middle of a human chromosome 2 that matched the 158 tandem repeats found in the telomeres of chimpanzee chromosomes 2A and 2B. The study revealed that at some point in human ancestry, the telomeres, or ends of the chromosomes 2A and 2B, fused head-to-head with one another. The probability of the independent origination of these matching sequences with no evolutionary relationship is highly unlikely, undermining the claims of evolutionary opponents.
Got some pretty strong evidence right from the start.
Yeah, but there's even more evidence for the fusion event. The sequence for centromere on 2A matches with the centromere for human chromosome 2, while the centromere on 2B aligns with a non-functioning sequence along the same chromosome. However, this non-functioning sequence has remnants of the alphoid sequences typical of a normal centromere, showing that perhaps there had once been a centromere in that place, the centromere of chromosome 2B. This further strengthens the case for our common ancestry with the great apes.
Another piece of evidence is shown by the high similarities of transposable element and retroelement sequences and positions of human and chimpanzee DNA. Numerous studies showed that the human and chimpanzee transposable element similarity was determined to be about 98%, while other great apes were similar with lesser varying degrees. Alu elements, the most common retroelement in the human genome, comprising of about 10%, serve as identifiable clues in determining our relationships with the great ape species.
In 1985, scientists at UC Davis and UC Berkeley discovered that humans and chimpanzees had Alu elements in the same exact places and orientations surrounding a set of hemoglobin genes. The DNA sequences of 7 Alu elements were 94.7 to 93.9% similar, indicating recent common ancestry that allowed minimal time for the small variations in sequence. The chance that 7 retroelements with their near-identical sequences and same position and orientation is very, very small, and therefore we can assume that humans and chimps have inherited these sequences from a very recent common ancestor.
A second example regarding Alu retroelements highlights the degrees of relatedness between humans, chimpanzees, gorillas, and orangutans. All 4 of these species have the CMT1A DNA segment, a gene that induces Charcot-Marie tooth disease through the duplication of a sequence between 2 copies of itself. Humans and chimpanzees are found to have 2 copies of the segment, with Alu retroelements flanking the ends of both copies. The CMT1A segments are in the same exact place, and both have a unique truncated version of an Alu element on the right side of the copy. Gorillas and orangutans only have 1 copy of the CMT1A segment, with flanking Alu retroelements, but in the same position as humans and chimpanzees, demonstrating that the duplication of the gene occurred after the common ancestor between the human and chimp group and the gorilla and orangutan group. We can see how Alu elements show degrees of relatedness across species and how scientists can analyze sequence, orientation, and position to create an evolutionary tree or cladogram.
Okay, now let's back up and compare the genomes of the chimpanzee and human.
In September of 2005, the Chimpanzee Sequencing and Analysis Consortium published the entire sequence of the chimpanzee genome, allowing scientists to delve deeper into the DNA comparisons between humans and chimps. Recently, scientists have developed the fluorescence in situ hybridization, or FISH method, which made remarkable progress with cross-species comparisons. First, scientists develop a probe, generally a single-stranded DNA fragment from one species, and add it to intact chromosomes of another species, in which the DNA was separated into single strands. Hybridization between the probe and the chromosome occur when highly similar pairs encounter one another. Chromosomes are illuminated with UV light, and the positions of matching DNA sequences are revealed through the fluorescent dye. Through the FISH method, scientists have discovered that more than 98% of human DNA sequences correspond with chimps, but that there is also 9 different rearrangements.
Although these rearrangements are significant differences between the human and chimpanzee genomes, they still support the evolution theory and mechanism of natural selection. The 9 inversions demonstrate that once chimpanzees and humans differentiated from one another, speciation was able to occur because of these chromosomal differences.
There is still more evidence to the theory, this time using pseudogenes to explain evolution. A pseudogene itself is a non-functional copy of a functional gene, having lost its use through multiple mutations. In 2009, a study done by scientists in the University of Victoria compared the pseudogene of the GBA gene between humans, chimpanzees, gorillas, orangutans, and squirrel monkeys. The results showed that the pseudogene existed with humans, chimpanzees, gorillas, and orangutans, but not the squirrel monkey.
It also revealed that 55 base pairs were deleted in the pseudogene of humans, chimpanzees, and gorillas. The likelihood of a gene having the same sequence deleted in 3 separate species is highly unlikely, supporting the idea of a common ancestor. The analysis of the pseudogene reveals that the lineage of monkeys split into that leading to the squirrel monkey, while the other would lead to the great apes and humans. Then once the pseudogene had been replicated, the lineage further split between orangutans and the lineage leading to humans, chimpanzees, and gorillas. This would explain why these 3 species would all have extremely similar pseudogenes.
This theory is backed up by a large amount of evidence. It's overwhelming.
I would have to admit that there is no denying that evolution exists. I mean, it's in our very own DNA.
Well, it seems that we were able to solve this question based on multiple pieces of evidence from scientists from all around the world. It seems that we really did solve this case. Thanks for joining us on this week's DNA Unsolved. Next week, we'll be back with a new question and more theories.
See you next time.